listening to The Truth Tank, and I am your host, The Tank. This is episode 31 of The Truth Tank. Happy Halloween to everybody in the US and around the world. In light of Halloween and to celebrate the occasion, I've decided to cover a scary topic. I haven't chosen haunted locations, monsters, or ghosts. I've chosen something that's equally as terrifying and probably more so, and that is bioweapons. Tonight's episode is the Halloween special and part one of a three-part series dedicated to looking at bioweapons and their use throughout human history and warfare. It's a pretty scary subject. It's probably one of the most terrifying and diabolical forms of warfare besides nuclear. So that got me thinking. I started thinking about bioweapons and their implications, and if they've been used in warfare on a large scale. So the more research I did, the more complex and in-depth the research got. So I've chosen to break this episode up into three parts. We'll be covering pretty much every aspect of bioweapons and their uses throughout human history. Right, so without further ado, let's get into tonight's show. This is episode 31 of The Truth Tank, and it's the first ever Halloween special. A History of Bioweapons, Part 1. A background to bioweapons and their use in warfare from the 1500s right down to the 1800s. So what is a bioweapon, aka germ warfare? Has one ever been used before? Is there any truth to bioweapons, or are they just a terrifying sounding word made up to drive a plot of an action film? Well, unfortunately, bioweapons are all too real, and they've been in use for thousands of years. Bioweapons have been in use as long as human beings have been fighting one another. It is one of the earliest forms of a weapon of mass destruction, and it's one of the most effective. A bioweapon falls into a category of 4 separate weapon types that will come under the weapons of mass destruction umbrella, and I'm sure you're probably more than aware of what a weapon of mass destruction is. Just thinking back to the start of the Iraq War, that term WMD was thrown around a lot. The 4 WMD types are nuclear, radiological, biological, and chemical. So more or less, a bioweapon uses viruses, biological toxins, bacteria or infectious substances, and organisms like insects, fungi, and other fun stuff to use as the destructive component of the weapon. So rather than having a bomb that relies on the gunpowder and the dynamite and whatever else is in it to explode, and the shrapnel and the explosive power, biological weapons rely on the bacteria, obviously. We'll be referring to the above mentioned components of a bioweapon as viruses for continuity's sake. So rather than having to say biological toxins, bacteria, infections, and substances and organisms like insects, fungi, or other stuff, I'll just use viruses as a blanket term for all of the above. So like any weapon, the purpose is pretty simple. It's to spread the bad stuff, kill or harm people, animals, and plant life. This usually happens as an act of war, or a nation, a rogue state, or extremist group being a bunch of fuckwits wanting to cause harm and terror through the spreading of a biological agent. So the unique thing about bioweapons compared to other forms of WMDs is that they're technically alive or contain a living replicating organism. Viruses, according to most biologists, aren't alive due to the fact that viruses can't grow. They're not made up of cells like humans or animals are. They don't all make their own energy, and they can't keep themselves stable. But they do replicate, spread, and they can adapt well to a new environment, which makes them very resilient and very hard to get rid of. As we see on the news every time we turn it on. There's more news about Corona, COVID, whatever you want to call it, and how it's mutated into a different form. Last year we just had normal COVID, now we've got Delta. The question endures, is a virus a living thing? It's a very interesting point because they act like a living organism but lack some of the finer details of organic life forms. A majority of biologists say that viruses act more like an android than a biological organism. I found that point really interesting. I don't know a hell of a lot about viruses, and I've never heard that term used to describe a virus before. So that got me thinking, if they act more like a biological organism rather than an organic one, could they be artificial or biomechanical in origin? So in other words, could a virus be programmed? And if so, by who or what? I mean, obviously viruses and bacteria have their place in the world like everything else. But if they act so mechanical in nature, then does that suggest that they've been created by something or engineered to act a certain way? And that also brings up the bigger question, is if they are having programmed, they've been programmed to kill, so more or less someone or something is playing God. The effects are always different depending on the individual or group of people, so someone could be playing God. Because computer viruses act a very similar way. They're obviously being programmed for a certain purpose, much the same as an organic virus. And on the other hand, you really have to admire the resilience of a virus, because they kind of live in this grey zone in the world. They can't replicate, but they are very adaptable to a new environment. After doing some research, I've changed my opinion of viruses and how they work. I kind of view them as biological synthetic organisms. But the question always remains, by who, why, and for what purpose? There's an interesting article I found called off of scientificamerican.com titled "Are Viruses Alive?" by Louis P. Villarreal. It's from 2008. Although viruses challenge our concept of what living means, they are vital members of the web of life. In the episode of the classic 50s television comedy The Honeymooners, Brooklyn bus driver Ralph Cramden loudly explains to his wife, "Alice, you know that I know how easy you get the virus. Half a century ago, even regular folks like the Cramdens had some knowledge of viruses, a microscopic bringers of disease, yet it's almost certain that they did not know exactly what a virus was. They were, and are, not alone." For about 100 years, the scientific community has repeatedly changed its collective mind over what viruses are. First seen as poisons, then as life forms, then biological chemicals, viruses today are thought of as being in a grey area between the living and non-living. Spooky. They cannot replicate on their own, but they can do so in truly living cells and can also affect the behaviour of their host profoundly. The categorization of viruses as non-living during much of the modern era. The categorization of viruses as non-living during much of the modern era of biological science has had an unintended consequence. It has led researchers to ignore viruses in the study of evolution. Finally, however, scientists are beginning to appreciate viruses as fundamental players in the history of life. It is easy to see why viruses have been difficult to pigeonhole. They seem to vary with each lens applied to examine them. The initial interest in viruses stemmed from their association with diseases. The word virus has its root in the Latin term for poison. In the 19th century, researchers realized that certain diseases, including rabies and foot and mouth, were caused by particles that seemed to behave like bacteria, but were much smaller. Because they were clearly biological themselves and could spread from one victim to another with obvious biological effects. Viruses were then thought to be the simplest of all living, gene-bearing life forms. The demotion to inert chemicals came after 1935, when Wendell M. Stanley and his colleagues at what is now the Rockefeller University in New York crystallized the virus, tobacco mosaic virus. That sounds lovely. Kind of sounds like a cigar or cigarette company. For the first time, they saw that it consisted of a package of complex biochemicals, but it lacked essential systems necessary for metabolic functions, the biomechanical activity of life. Stanley shared the 1946 Nobel Prize in Chemistry, not in physiology or medicine, for this work. Further research by Stanley and others established that a virus consists of nucleic acids, DNA or RNA. By that description, a virus seems more like a chemistry set than an organism. But when a virus enters a cell, called a host after infection, It is far from inactive. It sheds its coat, bears its genes, and induces these cells' own replication machinery to reproduce the intruder's DNA or RNA and manufacture more viral protein based on the instructions in the viral nucleic acid. Newly created viral bits assemble and voila, more viruses arise, which also may infect other cells. These behaviors are what led to many to think of viruses as existing at the border between chemistry and life. More poetically, virologist Mark H. V. Van Regenmotel of the University of Strasbourg in France and Brian W. J. Mahey of the Centers of Disease Control and Prevention have recently said that with their dependence on host cells, viruses lead a kind of borrowed life. Interestingly, even though biologists long favoured the view that viruses were mere boxes of chemicals, they took advantage of viral activity in the host cells to determine how nucleic acid codes for proteins. Indeed, modern molecular biology rests on the foundation of information gained through viruses. Molecular biologists went on to crystallize most of the essential components of the cells and are today accustomed to thinking about cellular constituents. For example, ribosomes, mitochondria, membranes, DNAs, and proteins, as either chemical machinery or the stuff that the machinery uses or produces. This exposure to multiple complex chemical structures that carry out the process of life is probably a reason that most molecular biologists do not spend a lot of time puzzling over whether viruses are alive. For them, the exercise might seem equivalent to pondering whether those individual subcellular constituents are alive on their own. This myopic view allows them to see only how viruses co-opt cells or cause disease. The more sweeping questions of viral contributions to the history of life on Earth, which I will address shortly, remains for the most part unanswered and unasked. The seemingly simple question of whether or not viruses are alive, which my students often ask, has probably defied a simple answer. All these years because it raises a fundamental issue: what exactly defines life? A precise scientific definition of life is an elusive thing, but most observers would agree that the life includes certain qualities in addition to the ability to replicate. For example, a living entity is in a state bounded by birth and death. Living organisms also are thought to require a degree of biomechanical autonomy, carrying on the metabolic activities that produce the molecules and energy needed to sustain the organism. This level of autonomy is essentially to most definitions. Makes you wonder about machines in the future with artificial intelligence. Is a machine alive if it can replicate itself, even if it just figures out how to operate machinery in a factory and assemble parts? Kind of like the supercomputer in Terminator. So if a machine makes a machine, does that constitute life, or is that just some abomination or perversion of life created by a biological living entity, aka humans? Viruses, however, parasitize essentially all biomolecular aspects of life. That is, they depend on the host cell for the raw materials and energy necessary for nucleic acid synthesis, protein synthesis, processing and transport, and all other biomechanical activities that allow the virus to multiply and spread. One might then conclude that even though these processes come under viral direction, viruses are simply not living parasitites of living metabolic systems. But a spectrum may exist between what is certainly alive and what is not. A rock is not alive, a metabolically active sac devoid of genetic material and the potential for propagation is also not alive. A bacterium, though, is alive, although it's a single cell. It can generate energy and the molecules needed to sustain itself, and it can reproduce. But what about a seed? A seed might not be considered alive, yet it has a potential for life, and it may be destroyed. In this regard, viruses resemble seeds more than they do live cells. They have a certain potential which can be snuffed out, but they do not attain the more autonomous state of life. Another way to think about life is an is an emergent property of a collection of certain non-living things. Both life and consequences are examples of emergent complex systems. They each require a critical level of complexity or interaction to achieve their respective states. A neuron by itself, or even by a network of nerves, is not conscious. Whole brain complexity is needed, yet even in an intact human brain can be biologically alive but incapable of consciousness, or brain dead. Similarly, neither cellular nor viral individual genes or proteins are by themselves alive. The unulcerated cell is akin to the state of being brain dead, in that it lacks a full critical complexity. A virus too fails to reach a critical complexity. So life itself is an emergent complex state, but it is made from the same fundamental physical building blocks that constitutes a virus. Approached from this perspective, viruses, though not fully alive, may be thought of as being more than inert matter. They verge on life. Alright, I think we'll leave that article there because it kind of goes on and on and on about the same stuff. Alright, probably starting to get the picture. So back to it. So already, bioweapons are shaping up to be pretty complicated and interesting. Later on the show, we'll be having a detailed look at the history of these weapons and see if they've ever been used during the wars or terrorist attacks. So the real obvious and horrifying aspect of biological weapons is what the effects would be if one was ever used, especially one that was used on a large scale. They are pretty terrifying to think of. I think nuclear warfare and biological warfare are probably the most scary. Well, then again, chemical weapons is pretty frightening too. All the weapons of mass destruction are equally as terrifying. So when you boil it down, weapons of mass destruction, whether it be nuclear, biological, radiological, or chemical, are all bad for business. They're all designed to wipe out life on Earth, and they don't give a fuck who's standing in the way. Everyone can be wiped out by one of these weapons, which makes you wonder why the fuck anyone wanted to build them in the first place. So why would someone or some nation or group want to use a biological weapon? Put simplistically, a bioweapon is a great way to gain a tactical or strategic advantage over an enemy. Whether this be in the conventional sense, such as a modern-day war, or an extremist group spreading fear, or a rogue nation or group tipping the balance of power for their own ends. There are several ways to achieve this outcome. One is obviously pretty simple, is to possess a biological weapon and to use it. The second way to use a biological weapon is far more deceptive and a hell of a lot sneakier. It is to threaten the use of a biological weapon by either a said group or individual, even if they do not have one or cannot even get their dirty little hands on one. The threat of having a weapon of mass destruction, even if you don't possess it, is nearly as frightening if someone has one and is intending to use it. The Hollywood version of this is for a bad guy or guys to get their hands on a biological weapon, usually in the form of a bomb or a biological sample in a petri dish or a test tube, and release it on an unsuspecting population. And then you cut to the hero, aka Tom Cruise, doing some death-defying stunt to get the canister or disarm the bomb before the bad guys figure out what's going on. This brings me to the next point: the form or the vessel in which the biological contaminant is housed. When I was younger and I first heard the term bioweapon, I pictured a bomb full of bacteria or viruses, literally like test tubes or petri dishes full of viruses and shit, stuffed into a conventional looking bomb that would have been fired at an enemy and it would have blown up and contaminated everything. But in reality, it could really take any form, such as a infected person or even an object. They're the most likely and realistic scenarios. Someone gets infected with something, he gets on a plane, and next thing you know, the entire world's got it. Just look back 2 years ago with COVID-19. Started in one place and spread all around the world in a matter of months. Not saying that it was a bioweapon, but we can't rule out the possibility that it might have been. And obviously the other forms bioweapons can take are the bomb itself or an aerosol, which would be the more theatrical version of spreading a biological contaminant. The advantage of a bioweapon is you can target a certain or small population or even a single person. by injecting them with a biological agent or targeting a village or the entire world. One of the most interesting aspects of a bioweapon is they can be used as a area denial weapon. In other words, if you don't want an opposing force taking or setting up camp in a certain area, a biological weapon is a good deterrent. The Japanese did this in World War II by poisoning water sources to prevent Allied forces from drinking, which in turn leads to dehydration and death in the heat of the Pacific Islands. They also poisoned over 1,000 wells in China, just so they could study the effects of cholera and typhoids. The big problem is, unlike a nuclear or chemical threat that is obviously more or less instantaneous in its effects, biological weapons may take days or weeks to be effective, which really isn't going to stop any advancing army from taking a strategic location right away. And there's also no guarantees that it's going to be effective against everybody, or that the enemy will figure it out and counter the biological agent. You never know, they might bring a cure with them, or have everybody immunized beforehand. Hypothetically, if that was to play out, you might stop a majority or a minority of the army from advancing, but this might take a couple of days. They might make it a couple of miles inland before it takes place. And that may have given them enough time to set up a base camp, set up medical facilities, and supply routes. So that might not always be the best tactic to use. In terms of poisoning an enemy's food or water supplies or sources, it is a pretty effective form of warfare in that sense. We will be looking at that in depth on the second episode. Unlike nuclear and other forms of warfare, biological and chemical weapons don't always have to be lethal. Like in World War I, if you didn't put a mask on during a mustard gas attack, you could die outright, but other times it didn't always have an instantaneous effect. If you did inhale some of the gas, it wasn't always fatal. On the other hand, if you did inhale, sometimes you weren't always affected immediately, and you could have lifelong or multiple year long or even decade long effects. Hitler was a runner in World War I. He got gassed once and he had decades long health problems because of it. I think he had some type of breathing problem from it, but he was also either fully blind or partially blind in one of his eyes. And it took about 10 years for his eyesight to recover just because of the mustard gas attack. You could have some severe health side effects due to the gas, and everyone's side effects were different. So, like I said, some people died outright, some people didn't have any immediate effect, but had a lifelong of consequences to deal with afterwards. Hitler went blind, other people had lung problems, other people had nervous system problems. It was all different depending on the individual. So this brings up the next question. One would think there would be some form of governmental or United Nations control, and you'd be right. No country is technically allowed to manufacture bioweapons or chemical weapons for that matter. There is some crossover between the two. Some chemical weapons have a biological or viral component that is used, therefore the laws and restrictions are pretty much the same. That's not to say that some rogue nation or extremist group couldn't or won't ignore UN treaties and just make their own. And that's not the scary part. A rogue nation might be able to pull it off. A terrorist or extremist group might be able to. If you had to guess, they probably wouldn't have the resources or infrastructure to make a weapon that would be that effective, especially on a large scale. The real scary thing is that a lot of militaries and governments have made, used or weaponized some of the worst viruses on Earth for the purposes of warfare. So the real scary thing is not that some group or a rogue nation could make a bioweapon and use it. The real scary thing is most of the nations on Earth have already made, tested and used bioweapons on small and large scales over the last 100 years. And that is the real scary thing, is it's already been done. I'm going to read a list of the varying biological agents used in the manufacturing of bioweapons. These are the ones that have been weaponized by the military or nations at some point in time and were deemed of military importance. So, sitting at the top of the bacterial bioagents list is anthrax. Most people have probably heard of it. It's a pretty nasty thing, and it has been used in bioweapons for a very long time. Some of the other nasty viruses to be used are cholera, diphtheria, dysentery, listeriosis, I don't know what that is, but it doesn't sound very nice, glanders, Brucellosis bovine, caprine, and porcine. I'm very bad at reading names out. I do apologise for that, so don't laugh too hard. One of the worst viruses to be weaponized is the plague. And given the already colourful history the plague has, it also makes you wonder who in their right fucking mind would think weaponizing the plague was a good idea. So besides viruses, there's also chlamydial bioagents that are used in bioweapons, one of which is psittacosis. Bioweapons also use rickettsial bioagents such as Q fever, Rocky Mountain spotting fever, and typhus, the human variety and the murine variety. Viral bioagents are also used, a majority of which are various forms of encephalitis. There's Eastern equine encephalitis, Venezuelan equine encephalitis, and Western equine encephalitis. So that one covers multiple regions. So just in case the Western one doesn't work, you can always use the Eastern one. Japanese bee encephalitis is also another version of the encephalitis virus. Marburg hemorrhagic fever, Rift Valley fever, smallpox, and yellow fever. They sound lovely. That brings us to biological toxins, which include abrin, bitolamine, toxins A through G, ricin, which anyone who is familiar with Breaking Bad will know that one, saxitoxin, Staphylococcus aureus, tetrodotoxin, trisethan, mycotoxin, or however you pronounce it, which brings us to last but not least, biological vectors, such as malaria, dengue fever, yellow fever, arbovirus, chikungunya, Xenophalis schepos, or oriental flea, plague, and marine typhus. That is some of the main biological components used in bioweapons. A lot of them have been approved and researched by the military at some point in time. So thankfully, the powers that be have seen how dangerous bioweapons are. In 1972, the Biological Weapons Convention was signed. It's also known as the Convention on the Prohibition of the Development, Production, and Stockpiling of Bacteriological, Biological, and Weapons on their destruction. So it's pretty much a biological weapon disarmament treaty banning the use and development, production, distribution, and stockpiling of said weapons. The treaty was signed into life in 1972 and became effective as of March 26, 1975. It was also the first time in history that a treaty banned the manufacturing of a whole class of weapon of mass destruction. I find it really hard to believe that it was only signed in 1970, and it wasn't signed after World War I, and especially after World War II. I think it's pretty alarming, and it's also very scary that the human mind not only has used these weapons in two major wars, but also used them again in Vietnam. So it pretty much took three major conflicts and a whole bunch of other events for any government to actually do anything about it. Thousands, if not millions, of people had to die before the powers that be around the world decided that this was an inhumane form of warfare. On the other hand, it is good that a decision was finally made to ban a weapon of mass destruction that was so horrifying. It's also disturbing because WMDs are still around. And I think it's also doubly disturbing that bio and chemical weapons are the only two out of the big four to be banned. With all the environmental stuff that's going on, you'd think nuclear weapons would be banned, or very close to being banned or restricted heavily. They are restricted, but they're still in production. But I guess on the other hand, they do have some practical applications, such as asteroid defense, I guess on the other hand, they do have some practical applications, such as defense against asteroids and aliens. What if the planet's overrun by aliens and they're just as susceptible to radiation as we are? I guess we have a backup. So there's a nearly identical treaty for the use and production of chemical weapons. The treaty has so far been very successful, and it has no end date, which is good because it prevents less than honest nations and states from stockpiling or producing new weapons with the looming end date to the treaty. 183 states have signed the treaty and agreed to the terms and conditions. Only a few countries either haven't signed the treaty or are sitting on the fence, or don't intend to sign the treaty one way or the other. So there are a few reasons for this. On the surface, it seems like they just want to do their own thing and they want to be the only country in the world that may possess a chemical or biological weapon. Maybe that gives them an edge over the bigger nations that have stockpiled nuclear weapons for the better part of 50 years. Or maybe they see that as a viable form of warfare still. Who really knows? But on the surface, it does make these countries look very bad, and it makes them look like they either have something to hide or they've got some nefarious desire to have these weapons. Other reasons they might not have signed the treaty is maybe they don't want to be told what to do by the superpowers of the world, or an alliance of nations that could be seen as ganging up or pressuring a smaller country into signing a treaty that they don't fully understand, or a treaty that doesn't really apply to them. Maybe they're just countries that have never had a biological weapons program to begin with. Maybe they never had the resources, money, or the interest to study it like some of the bigger countries have. And maybe they think that, well, this doesn't really apply to us, so why is everyone pressuring us into signing something that doesn't even apply to us? Another 4 states have signed the treaty but haven't ratified the terms and conditions, which leaves 10 states that haven't signed or agreed to the treaty at all. Really makes you wonder how bad a WMD has to be, or how bad the moral implications have to be before they get banned. Obviously, nuclear weapons are not morally corrupt enough to be banned yet, but biological and chemical are. There is also a big distinction between bio and chemical weapons and their use in warfare versus the use of nuclear/radiological weapons during conflict. The latter are considered conventional weapons, i.e. use in conventional warfare, which really does bring up the question, is a nuclear bomb is considered conventional so that it passes the WMD morality test, but a biological weapon doesn't. In other words, you can't infect somebody with smallpox, that's a big no-no, but nuking a city and obliterating all life and contaminating the environment is okay, not to mention the radioactive fallout that pollutes everything around for the next 2,000 years. So nuclear bombs get the tick of approval because, like I just mentioned, they're conventional and they're known for their conventional destructive powers, such as their overall destructive, explosive, kinetic, and incendiary powers, which as an explanation is pretty fucked, given there has been 2 examples of what a nuclear bomb can do to a city. There have been many examples of bioweapons being used in warfare going back thousands of years, but never in a large-scale modern city unless you want to count COVID. If you choose to believe that COVID-19 is a bioweapon, not saying it's right, not saying it's wrong. Could be. No one will ever really know. All types of WMDs are horrible and destructive and should be banned, but I suppose they do provide endless plot lines for action movies and Tom Clancy novels. So going back to the treaty, aka the BWC, the treaty is considered to be successful due to the formation of a strong global norm against bioweapons. It also states that the use of bioweapons would be, and this is a quote, repugnant to the conscience of mankind. Once again, how are nukes and radiological weapons better / not repugnant to the conscience of mankind? Aren't all weapons in a way, especially the WMDs? I'm not an expert on this obviously, but isn't a bioweapon in a very fucked up way the lesser of 4 evils? A lot of viruses and or bacteria, germs and other bad shit used in biological warfare can be cured or treated by vaccines, antibiotics or other medical treatments, generally speaking. The plague can be cured, so can smallpox, cholera, diphtheria, dysentery, and a lot of other bad shit. Nuclear wipes out everything. It poisons the surrounding land, and it makes it unlivable, which there is no cure for. Chemical weapons kill everything in their path and poison the water and food supplies, to which there is no instant fix. And I suppose from a strategic military standpoint, chemical weapons and biological weapons wipe out a population and pretty much leave the infrastructure standing. Where a nuclear bomb obliterates all infrastructure, and obviously you can't just go roll into a city you've just blown apart because it's radioactive. You can't set up camp at ground zero. But I guess the same is true for biological weapons and chemical weapons unless you have the PPE or the vaccinations to stop whatever it is you've decimated the population with. That's getting a little dark. So if anything, the treaty has proven to be a good blueprint for disarmament. It also provides a clear and workable plan that is proven to work. As of today, there is no state that has declared being in possession of biological weapons or has expressed an interest in obtaining them, or that their use in warfare is a legitimate one. There has been one roadblock with the treaty/the conventions, and that is the lack of overall support from institutional bodies. So there is no United Nations or World Health Organization regulation that monitors the treaty. No one country has taken on ownership of that. And that seems a little bit stupid, because if there's no institutional body or there's no nation or organization that monitors it, then how the fuck is anything supposed to get done? How do you actually know that nations are abiding by the treaty if there is no one checking up on it? You might have a less than honest nation claim that they're following the guidelines of the treaty, but really in reality they could have a secret biological weapons programs. They could also be planning on stockpiling weapons if nobody's watching. How does anybody really know who's being honest and who's not? This is made all the more difficult due to the fact that there is no formal organisation or authority that checks that nations, states or others are complying with the terms of the treaty. What could go wrong with that? I mean, what is the point of the treaty if no one checks upon it? Or there is no governing body enforcing the rules of the treaty? It's probably not an envious task for any country to put their hand up and say we're going to be in charge of this, because you have the whole argument that one country is trying to police everybody else. They may feel like they're being victimised or unfairly looked at by one nation over another. So the real reason why bioweapons are so dangerous and pose such a great risk to pretty much every corner of the planet comes down to a few key factors. One, they're easy to make and develop in comparison with other WMDs. Two, they're easier to store and take up a lot less room than nuclear, chemical and conventional weapons. The physical size is a lot smaller. Just imagine how many viruses can be housed with the same amount of space as compared to nuclear weapons. You get a lot more value from biological weapons than you do from nuclear. So 3, biological weapons pose a greater destructive capability, even greater than that of nuclear bombs. I'm not sure I buy that one, largely due to the fact that after a nuclear bomb goes off, everything is irradiated and on fire. But a virus or bacteria spreads further than radiation can. It also infects more people than that of the fallout from a nuclear bomb. On the other hand, if a bioweapon is used for military purposes, it wouldn't be as effective straight off the bat. It could take days to get the desired effects on a population, and there is no guarantee that your own troops, friendly forces, populations or places won't be affected as a result. So in a way, bioweapons are more risky because the outcome is a lot harder to predict, as opposed to a nuclear bomb, which there is plenty of data that can give us a pretty accurate indication as what the end results are going to be. So you're definitely rolling the dice when it comes to using a bioweapon, especially if it's a weaponized aerosol. The wind could change and blow it back into your face. As a tactical offensive weapon or a deterrent, bioweapons are very risky, even more so when compared to conventional weapons. Obviously that is just one scenario that could happen. A different delivery system gives you a different outcome. Same goes for the biological component, i.e. smallpox or anthrax. Then there's the systematical side of biological warfare to take into account. If a rogue nation, terrorist or extremist group could convincingly threaten a bioweapon attack, they can dictate the terms and conditions to said country. especially trade or commerce. So why would a nation want to do that, if it is even a realistic possibility? So hypothetically, if China was trying to release or have released a biological weapon onto the world, it could effectively allow them to hold or stall world trade. It also allowed China to catch up and manipulate world trade and commerce. It could also collapse world trade, force closures of jobs and industries, and create disorder and chaos around the world. It's hypothetical, of course, but just look at what's going on around the world. You have riots in Melbourne, you have the world's longest lockdown. Last year the world was plunged into chaos, the entire planet was pretty much locked down. Everybody's pissed off at each other, authorities are exercising their powers over a disobedient populace, the people are getting pissed off at the police for doing their jobs, governments are struggling to keep up with the changing situation, and overstepping their authority to the point that when things start to level off, they don't want to give up their power or control. Then there's the one set of rules for one group and another set for those with money. And the endless cycle of governmental corruption that COVID has made even more visible. Out of all the possible biological scenarios, there is one that is possibly the scariest and that could cause the most devastation on a global scale, and that is the escape from a lab theory. I don't quite know if it's quite the same as biological warfare, because accidents do happen. On the other hand, the escape from a lab theory does provide a very convenient excuse to let something out of a lab to run amok all over the world. It has happened before and will likely happen again. Even if you have state-of-the-art technology and facilities, accidents do happen. Labs blow up, mistakes are made even by those who research this stuff on a daily basis, and pathogens do escape. And unfortunately, it does happen before anyone realizes there's an issue. I found a very interesting article that relates to this on Vox.com, entitled How Deadly Pathogens Have Escaped a Lab Over and Over Again. Pretty eye-opening and alarming article, because all of it is true. It's by Kelsey Piper from March 20th, 2019. Research into dangerous viruses and bacteria is important, but for the deadliest pathogens, it's not clear the benefits are worth the risks. And that is a key takeaway from the article. In 1977, the last case of smallpox was diagnosed in the wild. The victim was Ali Mawal Malin of Somalia. The WHO tracked down every person he'd been in face-to-face contact with to vaccinate everyone at risk and find anyone who might have caught the virus already. Thankfully, they found no one had. Malin recovered and smallpox appeared to be over forever. That moment came at the end of a decades-long campaign to eradicate smallpox, a deadly infectious disease that killed about 30% of those who contracted it. Around 500 million people died of smallpox in the century before it was annihilated. But in 1978, the disease cropped back up, in Birmingham, in the United Kingdom. Janet Parker was a photographer at Birmingham Medical School. When she developed a horrifying rash, doctors initially brushed it off as chickenpox. After all, everyone knew that smallpox had been chased out of the world. Parker got worse and was admitted to hospital, where testing determined that she had smallpox after all. She died of it a few weeks later. How did she get a disease that was supposed to be eradicated? It turned out that the building that Parker worked in also contained a research laboratory, one of a handful where smallpox was studied by scientists who were trying to contribute to the eradication effort. Some papers reported that the lab was badly mismanaged, with important precautions ignored because of haste. The doctor who ran the lab died of suicide shortly after Parker was diagnosed. Somehow smallpox escaped the lab to infect an employee elsewhere in the building. Through sheer luck and a rapid response from health authorities, including a quarantine of more than 300 people, the deadly error didn't turn into an outright pandemic. It's also pretty lucky. That could have had the potential to go a lot worse. Imagine if that infected everybody in the hospital, or spread to everybody in Birmingham. It's a pretty horrifying thing to think of, but it can happen. Could something like that happen today? All over the world, bioresearch labs handle deadly pathogens, some with the potential to cause a pandemic. Sometimes researchers make pathogens even deadlier in the course of their research. That's nice of them. As Science magazine reported last month, the US government just approved 2 such experiments after years of keeping them on hold. I'm sure there's a good reason they do this, but it does bring up a hell of a lot of questions. I mean, obviously if it serves a good purpose, it might be worth the effort, but to make a horrific disease like smallpox deadlier, or anthrax or something like that deadlier, it does seem like you need to take a hell of a lot of precautions. You could also inadvertently create a super virus that doesn't really have a cure, or a virus where the current immunizations don't have much of an effect. So research into viruses can help us develop cures and understand disease progression. We can't do without the research, but on a few notable occasions, it's gone dangerously wrong and even killed people. Reviewing the incidents, it looks like there are many different points of failure. Machinery that's part of the contaminant process malfunctions, regulations aren't sufficient or aren't followed. Human error means live viruses are handled instead of dead ones. Yeah, the human error one is scary. I mean, if you had a bad day or you're tired, you go into the lab, you pick up the wrong vial, you misread it, it could be a recipe for disaster. Sometimes these errors could be deadly. If an enhanced novel strain of flu escaped from a laboratory and then went on to cause a pandemic, then causing millions of deaths is a serious risk. Mark Lipsick, a professor of epidemiology at Harvard, told me. If the past 2 years aren't a good example to go by, I don't know what is. It's not that there's a high risk of mistakes in these labs. The rate of mistake is actually quite low, but it's one thing to run a 1 in 1,000 chance of killing a handful of others from a mistake. The odds we face over a lifetime of driving. It's another thing entirely to accept a similar probability of killing millions of people. The cost-benefit analysis of pathogens which might kill the people exposed or a handful of others is vastly different from the cost-benefit analysis for pathogens which could cause a global pandemic, but our current procedures don't really account for that. As a result, we're running unacceptable risks with millions of lives. How pathogens can find their way out of the lab. The US government controls research into select agents and toxins that pose a serious threat to human health. From bubonic plague to anthrax, there are 66 select agents and toxins regulated under the programs, and nearly 300 labs approved to work with them. Researching pathogens and toxins allows us to develop vaccines, diagnostic tests, and treatments. New biology techniques also allow for more controversial forms of research, including making diseases more virulent or more deadly to anticipate how they might mutate in the wild. Okay, that's interesting. So this research can be really important and a critical part of public health efforts. Unfortunately, the facilities that do such work can also be plagued by a serious problem, human error. The 1978 smallpox death was, most analyses found, caused by carelessness, poor lab safety procedures, and badly designed ventilation. Most people would like to think that we are not so careless today, but scary accidents caused by human error, software failures, maintenance problems, and a combination all of the above are hardly a thing of the past. In 2014, as the Food and Drug Administration, the FDA, did cleanup for a planned move to a new office, hundreds of unclaimed virals of virus samples were found in a cardboard box. That's frightening. In the corner of a cold storage room, 6 of them, it turned out, were vials of smallpox. No one had been keeping track of them. No one knew they were there. They may have been there since the 1960s. That's funny and frightening. Just imagine if you accidentally took that home thinking it was junk, sticking it in the shed, having it break and spill out. It'd be a fucking nightmare. Or if you sold that at a garage sale, someone buys it, you put it up on Marketplace, someone comes and buys it, spreads it, and it goes from there. It's a recipe for disaster. Good plotline for a movie. Panicked scientists put the materials in a box, sealed it with clear packaging tape, and carried it to a supervisor's office. This is in brackets. This is not approved handling of dangerous biological materials. I'd hope not. It was later found that the integrity of one of the vials was compromised, luckily not one containing a deadly virus. In a lengthy report on how the incident happened, the FDA found persistent, horrifying shortcomings in the handlings of these incredibly dangerous materials. Among them, the security and inventory control of orphan biological materials whose owner departed the lab, but did not properly remove, destroy, or transfer the material to a new owner, was not maintained. FDA did not follow the CDC select agent guidelines for the packaging and transfer of samples to a high contaminant facility for securing the materials. FDA did not conduct a complete inventory for all of its laboratories and associated spaces when smallpox was eradicated in 1980, and all biological agents that cause smallpox were consolidated under the WHO Collaborating Center repositories at the CDC. The FDA also did not conduct a complete inventory when the Federal Select Agent Program was enacted in 2003. The blizzard of dangerous errors over only a few months in 2014 and the additional errors uncovered by subsequent investigations inspired the US government to change its practices. The government called on all labs that handle secure substances to immediately improve their inventory policies and review their procedures, and to provide written documentation that they'd done so. It launched government-wide reviews to better understand how to safely regulate pandemic pathogens. The FDA began providing better training and conducting periodic audits to make sure that the safety procedures that were ignored in this case are being followed. The 1979 and 2014 incidents grabbed attention because they involved smallpox, but incidents of unintended exposure to controlled biological agents are actually quite common. Hundreds of incidents occur every year, though not all involve potentially pandemic pathogens. In 2014, a researcher accidentally contaminated a vial of a fairly harmless bird flu with a far deadlier strain. The deadlier bird flu was then shipped across the country to a lab that didn't have authorization to handle such a dangerous virus, where it was used for research on chickens. The mistake was discovered when the Centers of Disease Control and Prevention conducted an extensive investigation into the aftermath of a different mistake, the potential exposure of 75 federal employees to live anthrax. After a lab that was supposed to inactivate the anthrax samples accidentally prepared active ones. It's a pretty big fuck up. Everyone makes mistakes, but some mistakes have some pretty drastic consequences. The CDC Select Agents and Toxins program requires that theft, loss, release causing an occupational exposure or release outside of primary biocontainment barriers of agents on its watch list be immediately reported. Between 2005 and 2012, the agency got 1,059 release reports, an average of an incident every few days. Here are a few examples. That is horrifying. It sounds like the entire field needs a huge shake-up. If the construction industry had a couple of accidents every day, it would probably be shut down. It's obviously it's a different set of circumstances, different barrel of fish, but that is a lot of fuck-ups to happen in a very short time. They're fuck-ups that can cause pandemics and kill a lot of people. In 2008, a sterilization device malfunctioned and unexpectedly opened, exposing a nearby unvaccinated worker to an undisclosed pathogen. And that's obviously just a fuck-up. There's not much you can do about that. In 2009, a new high-security bioresearch facility, rated to handle Ebola, smallpox, and other dangerous pathogens, had its decontamination shower fail. The pressurized chamber kept losing pressure, and the door back into the lab kept bursting open while the scientists leaned against it to try and keep it closed. Building engineers were eventually called to handle the chemical showers manually. In 2011, a worker at the lab that studied dangerous strains of bird flu found herself unable to shower after a construction contractor accidentally shut off the water. She removed her protective equipment and left without taking a decontamination shower. She was escorted to another building and showered there, but pathogens could have been released in the meantime. What can you do in that situation? She was probably fine, realistically. If she's wearing a protective suit, she was probably fine, but you have to follow protocol. Now, the vast majority of these mistakes never infect anyone, and while 1,059 is an eye-popping number of accidents, it actually reflects a fairly low rate of accidents. Working in a controlled biological agents lab is safe compared to many occupations like trucking or fishing. But a trucking or fishing accident will at worst kill a few dozen people, while a pandemic pathogen accident could potentially kill a few million. Considering the stakes and worst case scenarios involved, it's hard to look at these numbers and conclude that our precautions against disaster are sufficient. We have to work with these flu viruses. That is how we can understand them. Michael Osterholm, Director of the Center of Infectious Disease Research and Policy at the University of Minnesota, told Scientific American, after the string of 2014 contamination disasters. What's more important is we have to be able to do this safely. That's really the key piece. We don't want to stop this work. I mentioned him before in one of the shows I did on COVID. Challenges of safe handling of pathogens. Why is running a lab without such errors so hard? A look at the CDC's records of select agent contamination failures helps answer that question. Errors come from many directions. With worrying frequency, people handle live viruses thinking they've been given deactivated ones. Technology that's a critical part of the contaminant process can fail unexpectedly. It's not that there's a single problem, it's that there are so many that there are a part of a contaminant process and all of them have some small risk of failing. We can secure against showers depressurizing and sterilization equipment flying open when it malfunctions, but many other pieces of hardware are a critical part of contaminant measures, and they might have obscure malfunctions under the wrong conditions too. These problems don't just occur in the US. In the United Kingdom, a recent investigation found more than 40 mishaps at specialist laboratories between June 2015 and July 2017, amounting to one every 2 or 3 weeks. Beyond the breaches that spread infections were blunders that led to dengue virus, which kills 20,000 people worldwide each year, being posted by mistake, staff handling potentially lethal bacteria and fungi with inadequate protection, and one occasion where students at the University of West England unwittingly studied live meningitis causing germs, which they thought had been killed by heat treatment. I'm not sure it'd be cool or frightening. Imagine if one of these labs accidentally created a zombie virus and released it on the world, and all those sci-fi movies that have exactly the same scenario happen, it actually played out in reality. I think that'd be pretty horrifying. It also makes you hope that the people who work in these labs are ultra clean, or obsessive hand washers at least. Because imagine if you had some filthy fuck who just doesn't wash their hands, wipes the hands on their fucking clothes, doesn't care, is just a real fucking grotty slob. Imagine if they worked in one of these labs and just brought home this shit on a daily basis or a weekly basis and just spread it, and who could potentially spread viruses and diseases throughout the community. That would probably be more scary than a souped up zombie virus, if it actually exists. Severe acute respiratory syndrome, or SARS, had an outbreak in 2003. Since then it hasn't reoccurred in the wild, but there have been 6 separate incidents of it escaping the lab. 1 in Singapore, 1 in Taiwan, and 4 times at one lab in Beijing. It makes you wonder why that Beijing lab is still functioning. And this is a quote. These narratives of escaped pathogens have common themes, argued an analysis of biocontainment failures by medical historian Martin Furmansky in the Bulletin of the Atomic Scientists. There are unrecognised technical flaws in standard biocontainment, as demonstrated in the UK smallpox case. The first infection or index case happens in a person not working directly with the pathogen that infects him or her, as in the smallpox and SARS escape. Poor training of personnel and slack oversight of laboratory procedures negate policy efforts by national and international bodies to achieve biosecurity, as shown in the SARS and smallpox escapes. It's easy to see why problems are hard to address. Adding more rules for these handling pathogens won't help, if the people infected are usually not the ones handling the pathogens. Adding more federal and international regulations won't help if the regulations aren't consistently followed, and if there are still unrecognised technical flaws in the standards for biocontainment, how would we know until an accident made these flaws apparent? This is a worry that's recently back in the news because the US government has approved research aimed at making certain deadly influenza viruses more virulent. That is, making it easier for them to spread from person to person. The researchers involved want to learn more about transmissibility and virulence. In order to better equip us to combat these diseases, the labs conducting such research have taken steps to ensure their safety and reduce the risk of an outbreak. But have they reduced it enough? We imagine that when there's an accident, it's because a ventilation system fails or someone just forgot to do something, or that it's sort of avoidable, mechanical or human error, Lipsitch told me. Yet many of the recent failures don't fit the pattern. Rather, it was people doing something that they thought was the right thing and was neutralizing a dangerous pathogen by killing it. And in fact, they still had some dangerous pathogen or contamination with a dangerous pathogen. My concern is not really one of these people will do something that's foolish or reflects poor training. My concern is that there'll be a human error of the kind that's not really avoidable. Lipstitch does not think we should tighten standards for most research. He argues that our current approach, while its error rate will never be zero, it's a good balance of scientific and global health concerns with safety. But for the most dangerous pathogens, the ones with the potential to spark a global pandemic, he points out that the calculus doesn't hold. The influenza pandemic of 1918 killed 50 million people. Models of how influenza spreads suggest that an escape from contaminant by an influenza virus might not be contained in the local community where the incident occurred, as Birmingham's smallpox outbreak thankfully was. Is it sufficient for procedures to look alright on paper when the stakes are so high? Are there any labs in the world that we can confidently expect to be sufficiently careful with pathogens to never ever make mistakes that other labs have made thousands of times despite high standards of caution? So far, too much biodiversity policy has been reactive, tightening standards after something goes wrong. Given how badly things can go wrong, that's not good enough. It will be exceptionally challenging to make the labs safer, but when it comes to the risk of pathogens, we simply have to be up to the challenge. There's another article from Vox titled "A Russian Lab Containing Smallpox and Ebola Exploded" by Kelsey Piper, September 18, 2019. I'll just skim through some of this, it's pretty interesting. Russia State Research Center for Virology in the city of Koltsovo in Siberia has one of the largest collections of dangerous viruses anywhere in the world. During the Cold War, the lab developed biological weapons and defenses against them, and it reportedly stored dangerous strains of smallpox, anthrax, and Ebola, among others. According to the Russian independent media, the laboratory was undergoing repairs when a gas bottle exploded, sparking a 30 square meter fire that left one worker severely burned. Glass throughout the building was reportedly destroyed in the blast, and the fire reportedly spread through the building's ventilation system. The lab is one of two in the world known to still hold samples of smallpox, which was eradicated from the wild in 1977. The other is in the United States. Experts say that under certain circumstances an explosion could lead to the release of deadly pathogens. Part of the wave of the force of the explosion would carry it away from the site when it was first stored. Joseph Kam, an associate professor at the Stanley Ho Center for Emerging Infectious Diseases at the Chinese University of Hong Kong, told CNN. That said, storage procedures for deadly pathogens like smallpox are extremely strict. The city's mayor has stated that there is no threat to the general population, and a spokesperson for the center has said that there's no hazardous pathogens was stored in the room where the blast occurred. Of course, Russian public reports on safety incidents are not always accurate. Right, if you want to read the rest of that article, head on over to Vox. So they were obviously accidents and not biological weapon attacks, but it does make you wonder what if there was a cover-up, such as a country having a strain of the flu escape one of its labs which spreads and causes a pandemic all over the world. What if the accident is a very convenient excuse? Maybe it was just an accident that got out of hand, or it could be something else. Just saying, it's worth thinking about. So would that constitute or be considered a form of biological warfare? Covering up an accident also brings up a lot of questions. It breaches the trust with the public. To put things in perspective, let's have a look at how many lab accidents there have been in the last century. So the first known biosecurity accident happened in 1903 in the United States and involved Bacillus malei. So a worker became infected with the bacterium. She developed glanders while performing an autopsy on a guinea pig. She contracted it through a small wound on her finger. She survived. The next accident doesn't occur till 1932. It was also in the United States and involved the B virus. A man named William Brabner, he died of the infection after he was bitten by a rhesus monkey during research. B virus was named in his memory. The next accident happens in 1943, involving scrub typhus. A woman named Dora Rush accidentally pricked herself with a contaminated needle containing the virus and died. It's a while in between accidents. The next one happens in 1960, involving foot and mouth disease in Europe. So foot and mouth disease was accidentally released 13 times throughout European laboratories. The vaccines were produced, and I guess they got on top of it in 1960. It then happens again in 1993, which caused another outbreak of foot and mouth. Fast forward to 1966, another outbreak happens in the UK involving smallpox. A man named Tony McLean, he was a photographer with the medical school in Birmingham, contracted a mild case of smallpox. 12 years later, another medical photographer contracts smallpox, and that's the case mentioned in the article. So in 1967, the Marburg virus breaks out in Germany after a lab worker was exposed to it by African green monkeys, or tissue samples of African green monkeys. 32 people were sick, but I don't think anybody died. Next accident happens in 1971 in the Soviet Union, and it's smallpox again. The Soviet Union was testing biological weapons on an island in the Aral Sea. 10 people were infected with smallpox and 3 people died. There was another smallpox outbreak in 1972 in London. A 23-year-old lab assistant at another medical school. There's a lot of medical schools where smallpox breaks out, I've noticed from this. She contracted the virus while observing the harvesting of live smallpox viruses from eggs. So 1963 and again in 1977, there were varying outbreaks of various diseases in Nigeria, all through labs. There was a total of 936 cases and around 54 deaths. In 1976, Ebola breaks out in the UK. The infection came about by an accidental contaminated needle. In 1977 and 79, there was an influenza outbreak in the Soviet Union and in China. The H1N1 influenza virus reappeared circulating in humans in 1977. It looks like it escaped out of a lab and circulated between the two countries. There was a bit of controversy to this. Some people say it accidentally snuck out of the lab, and others say that it was the result of a human challenge trial, which is more or less a clinical trial for a vaccine that went horribly wrong. So in 1978, there's another smallpox outbreak in the United Kingdom. This is the incident mentioned in the article involving the death of Janet Parker after accidentally contracting smallpox. In 1978, foot and mouth disease broke out in the United States. There was a lab accident in the Plum Island Animal Disease Center. It infected animals and was released to animals outside of the center. So in 1979, there was an anthrax outbreak in the Soviet Union. Anthrax spores were accidentally released from a Soviet military research facility near the city of Slurdovox. I'm probably not saying that right, and my apologies to anyone from that area listening. The city is now known as Yekaterinburg. The outbreak claimed 100 lives. The exact figures are unknown. There's also been some controversy with the record keeping. It was denied by Soviet authorities. Medical records of the victims were removed to hide some violations of their Biological Weapons Convention, which had come into effect a few years earlier in 1975. The US authorities got involved in the case and determined that the incident was a result of aerosoled plumes of anthrax spores, which matched strains and samples from the nearby laboratory. It's also referred to as the biological Chernobyl. So in 1988, Marburg virus breaks out in the Soviet Union. A researcher by the name of Nikolai Yestovin infected himself lethally with the virus after accidentally pricking himself with a syringe he used to inoculate a guinea pig for guinea pigs. So in 1990, Marburg virus breaks out again in the Soviet Union due to a lab accident. It kills one worker. So nothing happens for a decade or so. In 2001, there's an anthrax attack in the United States. September 18, 2001, it's a week after September 11, letters containing anthrax were mailed to several Democratic senators. The attack killed 5 people and infected 17 others. I'll be covering that in the next episode. And this is followed by another anthrax attack a year later near Fort Detrick. And also in 2002, West Nile virus breaks out in the United States. 2 cases are reported. In 2002, there's an incident in Japan involving Anthroderma banhemii. I have no idea what that is, and there's not much information on it. So in 2003, and then again in 2008, SARS breaks out in Singapore. A 27-year-old student develops SARS-like symptoms, and an investigation found out that he was exposed to infected samples of coronavirus and SARS. In 2003, and again in 2012, SARS breaks out in Taiwan. In a similar set of circumstances, a 44-year-old scientist at the National Defense University in Taipei was confirmed to have SARS. He had been working and studying SARS in Taiwan. An investigation later revealed that he got the virus due to laboratory misconduct. 2004, SARS breaks out in China. 2 researchers at the Chinese Center of Disease Control and Prevention got the virus and spread the infection to about 6 other people. 2004-5, Ebola breaks out in Russia. A researcher at the Russian Biological Weapons Research Facility accidentally infected herself with a contaminated needle containing Ebola. So in 2004, foot and mouth disease springs up again in the United States after another incident at the Plum Island Animal Disease Center. In 2005, H2N2 influenza virus springs up in the United States and 17 other countries. The virus comes about due to a pretty big fuck-up in influenza being used in virus control kits that were distributed by Meridian Biosciences. They used a 1957 pandemic strain of the H2N2 influenza virus and included it in a routine testing kit that was sent to over 5,000 laboratories. The virus has been used as a positive control in the testing kits when prepared by Meridian Biosciences. In 2005 and then again in 2015, anthrax pops up in the US. The US Army Dugway Proving Ground mistakenly shipped live anthrax 74 times to dozens of different labs. In 2007, foot and mouth breaks out in the UK due to an accidental discharge of the virus FMD5 BF47 1860. It's from an animal lab in Breitbart. 2009 and 2012, a lab worker in Germany is accidentally exposed to Ebola. 2004, a man named Malcolm Castaban dies after accidental exposure to a strain of Yersin pestis, a bacterium that causes the plague. So in 2010, classical swine fever virus is detected in the US. 2010, in the US, cross-contamination in a laboratory causes an outbreak of cowpox. In 2011, in Australia, a scientist accidentally gets infected with dengue fever through a mosquito bite that they got in a laboratory. So 2012, anthrax pops up in the UK after the Animal and Plant Health Agency accidentally sent out live samples of anthrax. 2012, in the United States, a man named Richard Din died after being infected during vaccine research into Neisseria meningitis at a lab in San Francisco. So in 2013, once again in the US, H5N1 influenza. A researcher at the Center of Infectious Diseases research accidentally punctured his glove and hand with a needle loaded with the virus. The following year in 2014, 8 mice, some of which may possibly have been infected with SARS, H1N1 influenza, escaped from a laboratory at the University of North Carolina. In 2014, H5N1 influenza was accidentally shipped from lab to lab in the United States. Also in 2014, 75 personnel at the CDC's Roebohl campus were accidentally exposed to viable anthrax samples. Also in 2014 in the US, 6 vials containing viable smallpox from the 1950s labelled Virola were found at the FDA's White Oak campus. Also in 2014 in the US, highly toxic Burkholderia perseudomalal, or however you say it, accidentally escaped from a BLS-3 laboratory in Tulane National Primate Research Center in New Orleans. In 2014, Ebola breaks out in Sierra Leone after it infected an epidemiologist. 2014, 30-year-old lab worker in South Korea was infected with dengue through a needle injury. 2016, a researcher was infected with Zika virus in the lab in the United States. 2016, 30 members of staff were exposed to toxic Norcadia bacteria at the CSIRO's Black Mountain site in Canberra, Australia. In China, in 2016 and 17, cleaning staff at a hospital were accidentally infected with Brucella. 2018, in Hungary, a lab worker was accidentally infected with Ebola. In 2019, an gas explosion occurred at Vector in Russia, and that was mentioned in the article. The worker suffered third-degree burns and the blast blew out a few windows. No biohazard material was stored in the room where the explosion occurred. In 2019, a man named Emile Germain died from a variant of VCJD 10 years after pricking her thumb during an experiment with prion-infected mice. Yeah. And finally, in 2019, in China, an accident in the laboratory at the Lanzhou Veterinarian Research Institute caused 65 workers to become infected with bacillus. A later report from Reuters indicates that a further 6,620 residents in Lanzhou have been infected as of November 2020. And the city's local government is saying that the outbreak was caused by polluted waste gas from a nearby biopharmaceutical factory, which was carried by wind down to the Veterinary Research Institute, where the first cases were recorded in 2019. That sounds like absolute horseshit. If it is that easy for things to go wrong in a controlled lab setting with highly trained scientists, epidemiologists and the likes, just imagine what could possibly go wrong with a bunch of fuckhead terrorists training to replicate a pathogen and turn it into a weapon. So that brings to an end the whys, whats, wheres and the uses of bioweapons. Now we come down to the history side of the podcast. So how did this horrific idea come about? And what was the overall intention in developing bioweapons in the 20th century? Given what was already known at the time, why and who thought developing bioweapons was a good idea? We will now have a look at the history of bioweapons in human history, starting with Cortez at around 1500, going right through to the 1800s. So bioweapons have been used for as long as there have been people. All of history has numerous examples of bioweapons being used on populations, mostly during wars. Other examples of biological warfare have occurred during exploration and colonisation attempts. Diseases were spread by Europeans to native populations during contact. Sometimes it was by accident and was spread innocently enough. There obviously wasn't a lot of medical or scientific knowledge back then. The average person wouldn't have been thinking about viruses and smallpox and spreading of exotic diseases to native populations. All they would have been thinking about was creating a new life for themselves, getting as much gold as they could carry, or exploring new lands in the name of the crown. Other times, the spreading of disease and viruses was completely deliberate, like Cortez intentionally giving smallpox to the Aztecs in 1520, during his rape and pillaging of Mexico and South America. We will begin our look at bioweapons throughout history, starting from the year 1500. Around 500 or so years ago, European nations began to explore and colonise what at the time was known as the New World, and areas previously undiscovered to Europeans at least. Cortez set sail for Mexico on an unholy crusade to relieve the populations of their gold and precious materials, and to spread Christianity, all in the name of the Spanish Empire. Cortez didn't have many men with him, but what he did bring was millions of invisible enemies in the form of germs. During the meeting between Montezuma and Cortez, Montezuma invites Cortez and his men into the city of Tenochtitlan, receiving the Spanish as VIP guests. In turn, Cortez repays his hospitality by taking the king hostage. This event in turn spread smallpox through the city, decimating the population by around 40%. The city's population at the time was about 200,000. A year later, Cortez returns and takes the city with local warriors. The population had been ravaged by smallpox over the past year. A lot of younger people, children and the elderly, died outright. Others, such as adults and the young, were incapacitated to various degrees. The knock-on effects of smallpox were horrific. It wasn't just the outright virus that did a lot of damage, it was a lot of the knock-on effects. Those who weren't sick would have had to take care of members of the family who were sick. And that also runs the risk of them getting sick and the entire family dying without anybody to take care of them. There obviously wasn't any type of medical or hospital cover back then. Even if there was, the system wouldn't have been able to cope. This also meant that people couldn't tend the fields. They couldn't produce food. Corn would sit on the stalks and rot. No one would be there to pick it. Crops would be lost. So that meant the healthy and the sick now would get sicker. Most of the population's overall health would start to diminish. The city would have been plagued by famine as well as diseases. Lack of food also means that most healthy people's immune systems would have been weakened, which now means they're more susceptible to smallpox and other diseases. It also means that the healthy now have to care for the sick. They can't do their normal jobs, they can't upkeep the city. And as time goes on, as disease and famine spread, more and more people would have just given in to the Spanish. So there's a quote by an unknown person who was there in Tenochtitlan during the time of Cortez. And the quote goes, "The plague lasted for 70 days, striking everywhere in the city and killing a vast number of our people. Sores erupted on our faces, our breasts, our bellies. We were covered with agonizing sores from head to foot." There's another quote from a Franciscan monk in Cortez's crew. This quote might have come from Bishop de Luna or de Landa, I can't really remember. I mentioned him on a previous episode a couple of years ago, who perfectly illustrates the misery and decay that was occurring in the city at the time. As the Indians did not know the remedy of the disease, they died in heaps, like bedbugs. In many places it happened that everyone in a house died. And as it was impossible to bury the great number of dead, they pulled down the houses over them, so that their homes became their tombs. This monk paints a very vivid and hopeless picture. People being buried in their houses because there were so many dead is a pretty grisly end to a society. Rabies is another disease that has been used over the past few millennia as a weapon. Leonardo da Vinci thought of a way to use rabies as a weapon in the 1500s. He came up with a pretty vile concoction in the form of a bomb. The bomb had a mixture of several pretty gross elements, including arsenic, sulfur, toxic toads, tarantula venom, and saliva from rabies-affected dogs. Yeah, just imagine getting hit by that. It'd be fucking disgusting. If it didn't outrightly kill you from the smell, if you got it in your eyes or your mouth or into a wound, it'd be pretty horrific. You would probably be pretty sick or dying within a matter of hours or a day or two. Not much else is recorded from the 1500s on bioweapon use, so this takes us to the 1600s. In the 1650s, there was a Polish general by the name of Casimir Siemowicz. He came up with a very similar idea to Leonardo da Vinci. His idea was probably a bit more practical. Da Vinci often made a lot of things more complex than needed to be. The reason for this is he didn't want his ideas being used by the wrong people. He didn't want a lot of his weapons of war to be replicated by just anyone, especially by his enemies or the people who financed his work's enemies. He often made things very complex and did the designs backwards so no one could understand them apart from him, which is a very effective ploy to keep the war machine from getting out of control, because a lot of his ideas were probably pretty game-changing and pretty scary for the time. So is this why he added such exotic ingredients like tarantula venom? It also makes you wonder how Da Vinci knew about tarantulas. So Casimir's idea involved filling clay pots with rabid saliva, yuck, and then catapulting the pots onto enemy forces in the hopes that it would cause an epidemic of rabies. The effectiveness of this tactic is pretty questionable because you'd have to probably get it in your eyes or mouth for it to be effective, and that's not to say how long the saliva or the bacteria in the saliva lasted before it died or was ineffective. In 1675, the German and French forces came to an agreement not to use poison bullets during conflicts. So obviously this was a common trait of the day, is poisoning bullets. That may or may not work. Some bacteria might be affected by the heat of the gun. It might render the bacteria a bit useless. It also is a bit pointless when you think about it, because the chances of being killed by the outright bullet from the gun would probably kill you quicker than the bacteria would. And that's not to say that a lot of gunshots in that time ended up in infection anyway. Poisoning bullets might not be the most practical form of biological warfare. During the 1700s, the use of biological warfare continued and evolved into new forms of diabolical effectiveness. In 1710, the Russians and the Swedes went at it. The Russians flung plague-infected corpses over the wall of the city of Revel. This was also the last time plague corpses were used during warfare. We'll be coming back to flinging corpses over walls in the next couple of episodes. So this brings us down to the time of colonisation. During the colonisation of the New World, in particular North America, a more refined form of this tactic is used to spread disease to devastating effect. It also shows a new diabolicalness and malice. And as we will see, this becomes a standard tactic in the years following first contact and later colonization and expansion attempts. During 1763, the siege of Fort Pitt in Pennsylvania, Captain Simon Eckire, I think that's how you say his name, gave several smallpox-contaminated blankets and handkerchiefs in a small metal box to delegates of the Delawares during a parley from the siege. So the delegates of the Delawares were I think a coalition of tribes from the Delaware area. They'd been laying siege to Fort Pitt for some time. A parley was brokered, so Mr. Eckire seized the opportunity to spread smallpox through as many native tribes in the area as possible. This was deliberately done to spread the disease throughout the native population in the hopes that it would bring an end to the siege. A man by the name of William Trent was the mastermind of the plan and sent a bill to the British Army outlining the plan to spread smallpox among the native tribes. So, why is this bill important? It's important because the invoice was approved, which confirms that the British were aware of the plan and had approved the use of smallpox as a weapon. To complicate the situation further, the year before an outbreak of the pox killed around 100 people in Ohio between 1763 to 64. So it's not 100% certain if the deaths are a result of the contaminated blankets or from the previous year's outbreak that occurred naturally and had been reported to have been in the area. This tactic had been talked about and was by this time pretty widely used as an eradication tool of native populations all around the world. In fact, a month after the Fort Pitt incident, a British commander named Lloyd Geoffrey Amherst and Colonel Henry Bouquet had openly discussed using infected blankets to spread smallpox throughout native tribes in the area. There are 4 pretty vile examples in the form of letters between these two that show how callous and malevolent they were towards native peoples. It also shows how evil and simplistic the tactic was. I will be reading the letters verbatim as they were written back in the day, and I do apologise for any offence. So the letters in question are from June 29th, July 13th, 16th, and the 26th, 1763. Amherst wrote on July 16, 1763, P.S. You will do well to try to inoculate the Indians by means of blankets, as well as to try every other method that can serve to extirpate this execrable race. I should be very glad your scheme for hunting them down by dogs could take effect. Yeah, this guy is a evil piece of shit. It is a piece of history that I am reading. I don't agree with any of it, so I do apologise for any offence. Bouquet replies on July the 26th, 1763. I received yesterday your Excellency's letters of the 16th, with their enclosures. The signal for Indian messages and all your directions will be observed. A similar tactic was tried a few years later. In 1785, near Tunisia in Africa, during the siege of Lakali, diseased clothing was thrown into the city, much the same as dead bodies had been in the centuries before. The tactics were evolving and becoming safer for those throwing them, in a way. It also showed a refinement in the use of biological agents in war. Clothes are less obvious than a dead body just lying there. Anyone could unwillingly pick up an item of clothing and use it, not knowing it was contaminated by a disease like smallpox. It also shows a new level of diabolicalness and malice. I do like that word, diabolicalness. So I found a very interesting article on history.com titled, Did Colonists Give Infected Blankets to Native Americans as Biological Warfare? There's evidence that British colonists in 18th century America gave Native Americans smallpox-infected blankets at least once. But did it work? It's by Patrick J. Kiger. North American colonists' warfare against Native Americans often was horrifyingly brutal. But one method they appear to have used, perhaps just once, shocks even more than all the bloody slaughter. The gifting of blankets and linens contaminated with smallpox. The virus causes the disease that can inflict disfiguring scars, blindness and death. The tactic constitutes a crude form of biological warfare, but accounts of the colonists using it are actually scant. Colonial weaponizing of smallpox against Native Americans was first reported by 19th century historian Francis Parkman, who came across correspondence in which Sir Geoffrey Amherst, Commander-in-Chief of the British forces in North America in the early 1760s, he discussed its use with Colonel Henry Bouquet, a subordinate on the western frontier during the French and Indian War. For all the outrage the account has stirred over the years, there's only one clearly documented instance of a colonel attempting to spread smallpox during the war, and oddly Amherst probably didn't have anything to do with it. There's also no clear historical verdict on whether the biological attack even worked. Smallpox had spread at Fort Pitt. Early American historian Elizabeth Fenn of the University of Colorado Boulder lays out her theory on what happened in her 2000 article in the Journal of American History. In the late spring of 1763, Delaware, Cheyenne, and Mingo warriors, inspired by Ottawa war leader Pontiac, laid siege to Fort Pitt, an outpost at the confluence of the Allegheny and Monongahela rivers in present-day downtown Pittsburgh. The fort's commander, Captain Simon Eckire, reported in a June 16th message to his superior, Philadelphia-based Colonel Henry Bouquet, that the situation was dire, with local traders and colonists taking refuge inside the fort's walls. Ukiah wasn't afraid of his Native American adversaries. The fort's hospital had patients with smallpox, and Ukiah feared the disease might overwhelm the population inside the fort's cramped confines. Bouquet in turn passed along the news about the smallpox inside Fort Pitt to his own superior, Amherst. In a June 23 letter, in Amherst's July 7 response, he cold-bloodedly saw an opportunity in the disease outbreak. Could it not be contrived to send the smallpox among those disaffected tribes of Indians? We must on this occasion use every stratagem in our power to reduce them. On July 13th, Bouquet, who at the point was travelling across Pennsylvania with British reinforcements for Fort Pitt, responded to Amherst, promising that he would try to spread the disease to the Native Americans via contaminated blankets, taking care, however, not to get the disease himself. That tactic seemed to please Amherst, who wrote back in approval on July 16, urging him to spread smallpox as well as to try every other method that can serve to expedite this execrable race. It must have been very risky to try to spread smallpox through blankets and contaminated gifts and not get it yourself. It seems like you'd be rolling the dice, and that's if it actually works. What Amherst and Bouquet didn't know was that somebody at Fort Pitt had already thought of trying to infect the Native Americans with smallpox and had attempted to do it. William Trent, a trader, land speculator, and militia captain, wrote in his diary that on June 23rd, 2 Delaware emissaries had visited the fort and asked to hold talks the next day. At the meeting, after the Native American diplomats had tried unsuccessfully to persuade the British to abandon Fort Pitt, they asked for provisions of liquor for their return. The British complied and also gave them gifts, 2 blankets and a handkerchief, which had come from the smallpox ward. I hope it will have the desired effect, Trent wrote. Though it's not completely clear who perpetrated the biological welfare attack, documentary evidence points to Trent as the probable culprit. Also detailed in Fenn's 2000 article, the trader later submitted an invoice to the British military for purchasing 2 blankets and a silk handkerchief to replace in kind those which were taken from people in the hospital to convey the smallpox to the Indians. Eckire certified that the items were used to spread smallpox, which indicates that they may have been in on the attempt as well. British General Thomas Gage, who succeeded Amherst that year as Colonel Commander, eventually approved the payment. Typical merchant trying to get his money back from services rendered from the government. That's one documented case that we have, says Paul Kelton, a historian at Stony Brook University and author of 2 books on the role of epidemics in the European takeover of the Americas. It's not known whether Bouquet actually followed up on Amherst's letter and made additional attempts on his own to spread smallpox to the Native Americans, he says. It's also not clear whether or not the attempt at biological warfare had the intended effect. According to Fenn's article, the Native Americans around Fort Pitt were struck hard by smallpox in the spring and summer of 1763. We can't be sure, Kelton says. Around that time, we know that smallpox was circulating in the area, but the Native Americans could have come down with the disease by other means. Historian Philip Ranlett of Hunter College and author of a 2000 article on the smallpox blanket incident in Pennsylvania History, a journal of Mid-Atlantic Studies, also cast doubt. There is no evidence that this scheme worked, Ranlett says. The infection on the blankets was apparently old, so no one could catch smallpox from the blankets. Besides, the Indians just had smallpox. The smallpox that reached Fort Pitt had come from Indians, and anyone susceptible to smallpox had already had it. Interesting. Good point. I mean, there would be no guarantee. How long does a smallpox virus last on clothing? Is it even as contagious on clothing, or does it need to come from a human host first? The most important indignation that the scheme was a bust, Ranlett says, is that Trent would have bragged in his journal if the scheme had worked. He is silent as to what happened. Even if it didn't work, British officers' willingness to contemplate using smallpox against the Indians was a sign of their callousness. Even for that time period, it violated the civilized notions of war, says Kelton, who notes that disease kills indiscriminately. It would have killed women and children, not just warriors. But Kelton cautions against focusing too much on the smallpox blanket incident as a documented method of attack against Native Americans. He says the tactic, however callous and brutal, is only a small part of a larger story of brutality in the 1600s and 1700s. During this period, British forces tried to drive out Native Americans by cutting down their corn and burning their homes, turning them into refugees. In Kelton's view, that rendered them far more vulnerable to the ravages of disease than a pile of infected blankets. That's kind of very similar to what Cortez did to the Aztecs. And this didn't just happen in North America, this happened everywhere. And this brings us to the next case. There's a very similar conspiracy, or in this case it's alleged conspiracy by some historians. It involves Australian Aboriginals and a very similar plan to spread smallpox through communities throughout New South Wales. Australian Aboriginals to this day claim that an outbreak of smallpox in 1789 was deliberately spread by the British. If Fort Pitt and the plot by the commanders Amherst and Bouquet are anything to go by, it is more than likely that this is the case. There is only circumstantial evidence at this stage, but every theory has some truth to it. There isn't a paper trail like the Amherst and Bouquet case, or a defining event that had been recorded. In the 1980s, a man named Dr. Noel Butlin put forth a theory that smallpox could have been used as an exterminating agent. He isn't the only historian to suggest this. David Day in 1997 and Dr. John Lambert in 2000 have suggested very similar theories. Day has said there remains considerable circumstantial evidence to suggest that officers other than Philip, or perhaps convicts or soldiers, deliberately spread smallpox among Aborigines. The Philip in question is Arthur Phillip. He was a naval officer who was the founding governor of the colony of New South Wales. He was a very reliable and very capable man who knew what he was doing, and this is why he got the job. This is why he was in charge of the fleet and why he was the first founding governor. So during the arrival of the Third Fleet, there was a concern over resources and land. So maybe these two factors do play a part in this plan. Lambert also claims that there is strong circumstantial evidence that suggests the smallpox epidemic, which ravaged Aborigines in 1789, may have resulted from deliberate infection. Every theory has a counter-argument, and this one is no different. In 2002, a woman named Judy Campbell contradicted this by saying smallpox had not occurred in any members of the First Fleet. She also said that the fleet's only source of infectious material was that meant for inoculation against the pox. She also claims there isn't enough evidence to suggest Aboriginals had exposure to any of the infected materials, if there were any. Campbell has also offered another interesting theory to the origins of the smallpox outbreak. She claimed that transmission was more likely due to regular contact between Aboriginals and Indonesian fishermen. The fishermen came from the port of Makassar on the island of Sulawesi in the Indonesian archipelago. They're also referred to as Marquesians. It turns out the archipelago had always had smallpox, so could this be the source of the 1789 outbreak? Contact between the two groups is very likely. There's always been theories that the Indonesians and even the ancient Chinese knew about Australia long before Europeans did, and had a pretty elaborate trade network and relationship with the Aboriginals that lived in the top end of Australia. So the likely contact would have been with the northern Aboriginals, not those down lower in the country like where the New South Wales outbreak happened. And if there was an outbreak in the north, it might have spread, but may not have spread due to distance and lack of contact. The only really thing wrong with this theory, if there was contact with the Marquesians, it probably would have been with the communities of northern Australia. They may have got down further, but there would have probably been, well, there would have undoubtedly been some conflict with the British. I don't think they would have appreciated it too much. Another fleet of ships muscling in on their turf, it probably would have resulted in bloodshed and conflict pretty quickly. You can probably theorise that contact was more than likely to the top end of Australia and probably to some of the coastal communities along the northern tip of Queensland. That, however, might contradict the outbreak in New South Wales, because if there was an outbreak of smallpox in these communities, it probably would have been in the top end. I'm not sure it would have got down as far as New South Wales, but it could have happened. I'm not saying either one of these theories is right or wrong. So if there was a breakout in the north, it might have spread, but it may not have spread due to the distance and the lack of contact. Who's to say the affected group hadn't died out by then, or were just too incapacitated by sickness to move? She also points out that a later outbreak of smallpox in 1860 was directly linked to the same Marquesian fishermen who spread smallpox to the Aboriginal settlements. The Marquesian fishermen more than likely traded with them the same way they had been for generations, and could have unwittingly spread the disease to the unsuspecting Aboriginals. From there, the Aboriginals who fled the infectious areas may have spread smallpox via trade and family routes inadvertently. It's a very similar set of circumstances to what happened in North America. There is also a lot of back and forth with these theories. One person, usually an academic, comes up with a theory. Another academic who has never left the classroom tries to disprove it. The Marquesian theory is viewed as controversial based on lack of evidence. Others say that the smallpox couldn't survive the trip from England to Australia. If the first fleet did have any type of viral samples intended to be used as a bioweapon, some historians have suggested that it would have been killed off by the heat of the trip. I do call bullshit on that one, because viruses have survived much worse, and they could have infected someone on the ship. It may not have been that noticeable, they may not have even been sick or noticeably sick, and they could have transferred it that way. I mean, if Cortez could bring smallpox from Spain to Mexico, it's very possible that smallpox made its way from England to Australia. Another historian by the name of Christopher Warren in 2007 said that the smallpox would have still been usable, and it could have been used in the 1789 outbreak near Port Jackson, which was the intended site of the convict settlement. He reviewed his own theory a few years later, in 2013, and claimed that smallpox didn't spread through Australia before 1824. He also says there wasn't any smallpox in Marquesia, and that it couldn't have come from there and spread throughout the Aboriginal populations, and wasn't responsible for the Sydney outbreak. He does leave one crucial detail out, and that is, what if others joined the Marquesian fishermen from other islands or parts of Sulawesi? He settles for the British were the ones that had intentionally spread smallpox in Sydney in 1789, and not the fishermen. They did this, as he claims, out of necessity due to the adverse conditions faced by the First Fleet, such as low ammunition, food and provisions, supplies, among other things. Just to make things even more confusing, another academic, a professor of physiology, John Comudi, has added his 2 cents and says the smallpox outbreak was unlikely and far-fetched, and that the outbreak was more than likely chickenpox and not smallpox. How is that far-fetched? I mean, how is smallpox more far-fetched than chickenpox? One is as bad as the other in an unimmunized indigenous population, especially one that has had no contact with smallpox or chickenpox before European settlement. So to round out the 1700s, the legendary French general Napoleon tried his hand at biological warfare, by flooding the fields around the Italian city of Mantua in the hopes that this stagnant water would spread malaria. So obviously he intended for mosquitoes to breed in the water and spread a disease. Only one problem with that would probably take quite a while to achieve. That's if the water didn't naturally dry up or soak into the ground or run off somewhere. Seems a bit risky and a bit inefficient, but worth a try, I guess. There was one ray of light, however, to close out the 1700s. In 1796, British doctor Edward Jenner developed the world's first vaccine against the disease, and that was the smallpox vaccine. He figured out that being infected with a milder cowpox virus gave a decent degree of immunity to the dreaded smallpox. This is also a natural disease that ended up acting as a naturally occurring vaccine. So in other words, the cycle is complete. Everything that occurs in nature has a cure in nature. So here's a fun fact. The word vaccine is Latin for cow, derived from the origins of the cowpox vaccine. The natural vaccine was used right up until the modern smallpox reared its ugly head in the 20th century. The World Health Organization eradicated smallpox from 1958 to 1977 in a worldwide vaccination program, making it the only human disease to be completely eradicated. That's very surprising that smallpox is the only one to be eradicated. I guess there's no money in it for pharmaceutical companies. The smallpox vaccine is still in production as a safeguard against biological warfare if it were ever to be used, bioterrorism, and against monkeypox, which is also transferable to humans. A man named Alan Watt Downey demonstrated that in 1939 that the modern smallpox vaccine was serologically distant from cowpox, and Vaccina was subsequently recognized as a separate viral species whose genome sequencing had revealed that Vaccina is most closely related to horsepox, and the cowpox strains found in Great Britain are at least closely related to Vaccina. That brings us to the 1800s and to the end of the show and part 1. So next episode we're going to be focusing on the 1800s, the 1900s and the modern day. We'll be having a look at biological warfare in use during World War I and World War II, and some of the biological attacks that have happened in the 20th century. That will be followed by part 3 with biological weapons in ancient warfare. So that's the end of the show, everybody. Thank you very much to everyone who listened. Happy Halloween to everyone out there. If you'd like to support the Truth Tank, there's a few things you can do. Subscribe to the Truth Tank on Apple, Podbean, Google, Spotify, wherever you get your podcasts from. Rate and review the show. Tell your friends, tell your family, tell people on the street, strangers on the bus. Help spread the show and get the word out there. I appreciate all the downloads and all the listens. I've got some big plans coming for 2022, so I appreciate all your support in growing the show. Until then, a big thank you to everyone who downloaded and listened. Until next time, I'm the Tank. This is the Truth. May the truth be with you.
Stop.